This protocol is a very simple and efficient way to detect mutants generated by CRISPR-Cas9 technology from numerous individuals.
The two main advantages of this technique are that it can be done in a few hours and that it is applicable to a multitude of organisms.
Primer design can be challenging for certain genes, and multiple rounds of primer design and verification may be required to make HRMA efficient.
To perform a gradient PCR, start by preparing a master mix. Then remove 19 microliters of the master mix for the non-template control, or NTC, in a 96-well plate. Then add the template to the remaining master mix. Aliquot 20 microliters of the sample mix into the 96-well plate. Perform PCR following the cycling parameters. And then generate the thermal melt profiles using the parameters mentioned in the manuscript. After gathering all the required material, prepare a master mix with 0.5 microliters of the DNA release reagent and 20 microliters of dilution buffer per individual to be genotyped. Using a multi-channel pipette, aliquot 20 microliters of the mix into a PCR plate, and leave the PCR plate on ice. Next, place the anesthetized G1 mosquitoes in a glass petri dish to keep them sedated, and place 8 wild-type mosquitoes in the second petri dish. Wipe a pair of tweezers with 70% ethanol, and use the disinfected tweezers to remove one of the mosquito's hind legs. Then submerge the leg in the DNA release reagent solution. Place the mosquito in the corresponding vial,